Hello friends, welcome back to our channel NEET Biology Lectures in English. I am Dr. Parveen, your biology professor. In this lecture series, we are talking about class 12 biology subjects, that is unit 1 reproduction. So in this particular lecture, we are going to study about methods of reproduction, particularly asexual method of reproduction in detail. First, what is reproduction? Reproduction means giving birth to young ones or producing offspring is called reproduction. Reproduction can be classified broadly into two categories, asexual reproduction and sexual reproduction. Here, asexual reproduction means A means absent. There is no sexual contact between a male or a female organism of opposite sex and only one parent will be involved in the production of their offsprings. So here gametes may be formed or may not be formed. So that is asexual method of reproduction. Whereas sexual reproduction means two parents of opposite sex, male and female, will be involved in the production of the gametes. Okay. Whereas sexual reproduction means two parents of the opposite sex are involved and there will be a gamete formation. Here you should notice what is gamete. Gamete means, for example, take a human being. The reproductive cell is called gamete. We have male and female two sexes. The female reproductive cell is called ovum and the male reproductive cell is called sperm. Okay, both these cells are reproductive cells and they are haploid in number. They have half the number of chromosomes. When they unite together, the zygote will have the 23 pairs of chromosomes. Okay, 46 chromosomes will be there for the zygote. So that reproductive cell of any organism is called gamete. So in asexual reproduction, gamete may or may not be formed, but in sexual reproduction, always gamete will be formed. So first let us see in detail about asexual reproduction. Asexual reproduction means a single individual or a parent is capable of producing the offsprings. Here the offsprings are identical to the parent. That is very important. So whatever the daughter cells produced from the mother cell through asexual reproduction, it will be 100% identical to the parent cell. That we call them as clones. Clones means exactly the same copy, like a Xerox copy. Here the clones or the daughter cells will be both morphologically and genetically identical. Morphological means the external appearance. Not only by the external appearance, at the chromosomal level, at the genetic level also, it will be exactly the copies of the mother cell. So that is an important thing in the asexual method of reproduction. Let us see which organisms reproduce by this method. Single cell organisms like bacteria, yeast, amoeba, all the single cell organisms, mostly they reproduce by asexual method of reproduction. Why? Because they are already single cell. They don't have any extra reproductive organs. All the essential functions of the body, the reproductive functions, everything will be carried out by the single cell only. Okay, so for them single cell is the complete unit of action. So these organisms, they reproduce by asexual method of reproduction. Not only that, some plants and animals also reproduce by asexually under some conditions. For example, see this picture. This is called Komodo dragon. It is a higher cellular animal. They reproduce both asexually and sexually. Asexually, this dragon reproduces by a method called parthenogenesis. That means only the female dragon from the ovum, the unfertilized ovum, it develops into an young one. This is called parthenogenesis. Here there is no male involved, only the female will be involved in the reproductive process. So at certain time, this organism also reproduce by asexual method. Most of the kingdom Protista you might have studied 5 kingdom concept, right? Kingdom Animalia, Plantae, Fungi, Protista and Monera. Kingdom Protista includes all the protozoans, single cellular protozoans. All of them reproduce by asexual method of reproduction. Not only Protista, Kingdom Monera. Kingdom Monera includes all types of bacteria. They all reproduce by asexual method of reproduction. Well, there are many types of asexual method of reproduction. See here, fission, budding, fragmentation, sporulation, vegetative reproduction, gemule formation, regeneration and agametogenesis. All these methods are asexual mode of reproduction only. Okay, so we will study everything in detail one by one. So in this particular lecture, let us concentrate on the first one, fission. What is fission? Fission means separation, dividing or splitting, like this you could say. Here the parent body divides into two or more identical daughter individuals. That is called fission. So this fission basically we can classify into four types: binary fission, multiple fission, sporulation and strobulation. So let us see first binary fission. What is the meaning for this word bi? Bi means two. Fission means division. Here one cell gets divided into two equal halves. That is called binary fission. What happens? The parent organism divides into two equal halves. So each half now becomes a daughter cell, individual daughter cell. So that is binary fission. So during this process, the nucleus, they divide mitotically or amitotically. That is called karyokinesis. Karyon means nucleus. We know karyon, nucleus. That's why we call prokaryotes, false nucleus, eukaryotes, true nucleus. Karyon means nucleus. First, during binary fission, the nucleus gets separated. That is called karyokinesis. The second step, the cytoplasm gets separated. That is called cytokinesis. Okay. See, let us see this example. This is a dividing cell. So in this cell, this would be the first cell. For example, here first what would have happened? Here first the nucleus would have divided. That is called karyokinesis. So once the nucleus gets separated, the cell membrane and the cytoplasm, here they start dividing. This is called cytokinesis. The cytoplasm gets separated and after some time two daughter cells will be arrived from a single mother cell. So this is how binary fission occurs. But this binary fission is also having four types. Okay. So what are the four types? Simple irregular binary fission, transverse binary fission, longitudinal binary fission and oblique binary fission. How we are dividing these types of binary fission? This is based on the plane of division. That means when a cell gets divided into two cells, at which plane or which direction or which angle it gets divided. That is called plane of division. For example, see this picture, this is a spring onion. This spring onion we can cut at any directions or any planes. Okay, we can cut like this, long or a small squares or a thin pieces or thin fibers like shreds, or an oblique angle like this. A one small onion or a one apple or one vegetable, we could cut this at different planes, right? Depending upon the plane, the shape also varies, right? This is called plane of cutting. Similarly, when an organism reproduces, at which angle or at which plane it reproduces, depending upon that, we are classifying this binary fission into four basic categories. So the first one is simple irregular binary fission. Example for this is amoeba. Why? Because amoeba has no definite shape. It is very much irregular organism. So these structures are called pseudopodia. So these are the false legs. They have some jelly like structure. They can move at any direction they want, like that, very much irregular. So in this amoeba, you cannot see where the division will start or which angle the plane of division starts. It's very hard to observe. So what will happen during cell division of an amoeba? First, the nucleus, they gets elongated. That means karyokinesis first occurs. Okay. After that, the cytoplasm divides. The cell division occurs, all the contractile vacuoles which are present inside the amoeba, it will get disappeared. Okay, the nucleus divide mitotically, then the cytoplasm divides. That means cytokinesis occurs. And after cytokinesis, two daughter cells will be formed and they get separated. So that's why a single amoeba is reproduced into two daughter cells by binary fission. Here it is very difficult for us to judge where the cell division starts, at which angle, because it is very much irregular in shape. That's why we call amoeba binary fission as an example for simple irregular binary fission. The second type of binary fission is transverse binary fission. So example for this are paramecium and planaria. Paramecium is a protista, protozoa. It lives, you can see this in stagnant water and free water, like this water environment. And the appearance of this organism is like a sole of a shoe. Okay, the shape is like this, like the sole of the shoe. Planaria. Planaria is a flat worm. It's a free living platyhelminthes. It is a single cell worm. So these organisms, they reproduce by transverse method of binary fission. What is transverse method? For example, if this is the organism, the cut will be in the middle like this. Okay. From that, two organisms will come. So this is called transverse binary fission. Let us see paramecium. See here, this is a paramecium and it has two nucleus, a micronucleus and a macronucleus. Whenever it starts reproduction, first, as we told, nucleus karyokinesis occurs. Okay, the nucleus divides and later that the cytoplasm divides. Here a groove is formed. So from that groove part only the cell gets separated. So that's why a groove formed here, and from a single cell two daughter paramecium will form. Okay, this is an example for transverse method of binary fission. The third one is longitudinal binary fission. So example for this is Euglena and Vorticella. Euglena is a single cell eukaryotes with flagella. It's a flagellate. You could see this in freshwater and saltwater environment. And Vorticella is a single cell organism. Look at this structure. This is like an inverted bell-like appearance. Okay. So these organisms, they reproduce by longitudinal binary fission. So longitudinal means the cutting of this division, cell division will be in a longitudinal plane. Okay. That's why this is called longitudinal binary fission. For example, see this picture. This is Euglena with flagella. What is flagella? Flagella is the locomotory organ for the organism. So like bacteria has flagella, this Euglena is also a flagellate. So during cell division, what happens? The nucleoli get disintegrated and nuclear body, it gets separated. The nucleus, it undergoes karyokinesis. Okay, nuclear division occurs at the same time. Here you could see there is a longitudinal furrow. A cut or a groove occurs. In that point, it gets separated into two daughter nuclei. Also, you could see here, similar to the parent Euglena, both the daughter cells have individual flagella. So, during cell division, in this asexual reproduction, whatever the cell components are present, it is completely equally distributed to the daughter cells. The fourth method of reproduction is oblique binary fission. So, example for these are dinoflagellates. Dinoflagellates are small microorganisms which we could see in water bodies. Okay. So, one example, one species is Serratium. They do reproduce by oblique method of binary fission. So what happens here, the plane of division is at an oblique angle like this. You see, it is not transverse, it is not straight, it is in an oblique angle. So that's why we call this as an oblique binary fission example. This dinoflagellates, I told you, we could see this in some seashores, some oceans and all. They have these characteristics of bioluminescence. Okay, so during night they emit this bioluminescence, which will be very beautiful to see. Even in some Michael Jackson movies and all, you would have seen this bioluminescence during his song. Okay, and I think Life of Pi, in the movie also we could see this bioluminescence on the ocean. Okay, so some example for the bioluminescence organisms are dinoflagellates. Here, binary fission means the first organism which comes to our mind is bacteria, because 100% of bacteria, they reproduce by binary fission only. So if I ask you, can you tell whether a bacteria is a male or a female? There is no sex in bacteria, right? There is no male bacteria or female bacteria. It is a single cellular organisms. So when they reproduce, we call that reproducing cell as a mother cell and the young ones as a daughter cells. This is a general term we used to say. Okay. Suppose, for example, if there is a mother cell, it wants to reproduce. What happens? First, the cell gets elongated. After elongation, what happens? The DNA gets segregated, because inside the cytoplasm of the bacteria, it's full of DNA. Okay, so this DNA gets segregated. Karyokinesis again. Okay, so once this karyokinesis occurs, the next step is cell division, cytokinesis. So there will be a division of the cell wall and the cell membrane. And finally, two complete daughter cells will evolve. So like this, from one mother cell, daughter cell, two daughter cells will evolve. So similarly, if some 10 bacteria are present in a particular place, after many rounds of cell division, in one day we could see millions of bacteria, because one bacteria give rise to 2, 2 give rise to 4, 4 to 8, 8 to 16, like this exponentially it grows. Okay, that's why bacterial cell division is very faster compared to any other microbial cell division. Okay, the second method of fission is multiple fission. So here what happens, previously we have seen binary fission. In one cell division, two daughter cells are formed. Here, in one cell division, many daughter cells are formed. That is called multiple fission. So example for multiple fission is Plasmodium. Plasmodium is a parasite which causes malaria, malarial disease, you know, right? So this malarial parasite, it has a two-stage of life cycle. One stage inside the human being, inside the human body. The another stage inside the mosquito. Okay, so they have two stages during their life cycle. So they are called schizont and oocyst. Schizont is the stage inside the human beings. It is the reproductive stage. Similarly, oocyst is the reproductive stage inside the mosquito body. Right. So what happens during multiple fission? For example, if this is a schizont, a stage inside the human cell, reproductive stage means this Plasmodium schizont stage initially first has one nucleus surrounded by a cell, cytoplasm cell membrane. Suddenly, they produces many nucleus, multiple nucleus will be produced throughout the cell body. Okay, and after some period of time, this nuclei, daughter nuclei are surrounded by the outer covering. So they are called merozoites. And finally, these small daughter cells, they rupture the schizont cell wall and gets released out. Right? Here you could see the difference between binary fission and multiple fission. In binary fission, for example, like in bacteria, in amoeba, what happens? The mother cell is there. Mother cell is not getting ruptured or not getting died. Okay, it gets splitted only. That's why one cell becomes two cell. But here in the multiple fission, the mother cell is there, inside many nucleus and many sporozoites are formed. They rupture the mother cell and get released out. This is a method of reproduction in Plasmodium. As I told you, this Plasmodium has two stages. If this stage occurs inside a human being, the schizoid stage, the daughter cells are called metrozoites. So this process is called schizogony. Similarly, if this process occurs inside a mosquito, that is in a oocyst stage, sporozoites will come. The daughter cells are called sporozoites and this process is called sporogony. So two different stages occurs in two different organisms of the life cycle of a Plasmodium and we call them as a two different names. And another example for multiple fission is again amoeba. So amoeba has many types of reproduction. Okay, so another method of reproduction is multiple fission. So what happens when the environment is favorable for an amoeba? It reproduces by binary fission. One cell divides into two cells. But when the environment is unfavorable, what happens? This amoeba, it withdraws all its pseudopodia. These are the pseudopodia of the amoeba, right? So the false legs. It withdraws all its pseudopodia and it forms a three-layered covering stage called cyst. Okay, so for any organism, what is important? Its nucleus is important because it carries the genetic information of the organisms. So in order for the organism to protect its nucleus, here the amoeba what is doing? This nucleus is covered by a three-layered cyst wall, very thick cyst wall. Okay, this stage is called encystation. So when the environment is unfavorable, when there is no food or no favorable temperature, the amoeba vegetative cell now turns into a cyst stage, right? Again, when the environment gets favorable, what happens? Amoeba, the nucleus suddenly become multiplied. Very fastly, the daughter nucleus, more and more number of daughter nuclei will be produced inside the cyst and it will be surrounded by an outer coating. So this structure is called pseudopodiospores and they rupture the cyst wall and many daughter nuclei comes out. Daughter amoeba comes out. Okay. So from one amoeba, many amoeba will come out. This is by a method called multiple fission, right? This stage occurs in amoeba during unfavorable environment. The third method of asexual reproduction is by sporulation. Spore means it's like some resting stage. These spores will be formed by some organisms during unfavorable environment. Particularly, take bacteria as an example. If the bacteria has a good environment, it will be actively multiplying by binary fission in its vegetative state. When the environment temperature is high or low, when there is no food, the bacteria shrinks its body and it turns out into a structure called spore. That is a resting stage for bacteria. Okay, but some organisms, they do reproduce during this spore stage. So it is a method of reproduction asexually. So sporulation can occur in favorable environment and also in unfavorable environment in some organisms. So example, Chlamydomonas, amoeba, again amoeba, and Penicillium. Chlamydomonas is a single cell algae which lives in stagnant water and damp soil. Amoeba, Penicillium is a fungi. So these are the examples for sporulating organisms. So this Chlamydomonas, this single cellular algae, they do reproduce by sporulation when the environment is favorable. This you must understand here. Watch keenly. Some organisms do reproduce by sporulation during unfavorable condition like amoeba. Some organisms do reproduce by sporulation during favorable conditions like Chlamydomonas, even Penicillium. Okay. So here Chlamydomonas, in the mother cell, many daughter nuclei will be formed and it will be surrounded by the cytoplasmic and other contents and they rupture the mother cell and releases out. The spores of the Chlamydomonas are called as zoospores. Okay. And sporulation is also a major method of reproduction in fungi. Penicillium. You must be knowing penicillium, right? We get the antibiotic penicillin from this Penicillium notatum or Penicillium chrysogenum fungus. Okay. This penicillium, if this is the body of the penicillium, they are called phyllates. Okay. In this phyllates, they produce chain-like spores. They are called conidia. This chain-like spores are called conidiospores. Here, at the base, these spores are young. And when it goes higher and higher, the spores are mature. Okay. These spores get released out, and wherever they get dispersed from their new organisms, new penicillin grows. So, this is a method of reproduction in fungi, penicillin. Fungi produces many types of spores. Particularly, penicillin produces conidiospores, that is chains of spores. Again, very good example for sporulation during unfavorable environment is amoeba. See, previously we have seen the amoeba reproduce by multiple fission when the environment is unfavorable. Okay, here when the environment is unfavorable, there is no cyst formation. No cyst wall, nothing. Sometimes what amoeba does, all this nucleus get disintegrate and chromatin blocks are formed. Okay, then this nuclei is surrounded by a outer covering. This structure is called spore. So inside one amoeba cell, many spore cells, these are the resting stage of the amoeba. Suddenly these spore stage are produced. Again, when the environment is favorable, these spores get released out and they develop into a vegetative young amoeba cell. Okay. So, in amoeba, we could see 4 types of reproduction. Binary fission, multiple fission, sporulation and regeneration. And these condition depends upon whether the organism is facing favorable environment or unfavorable environment. It depends upon their living condition. Okay. So, 4 methods of reproduction amoeba does. The 4th method of reproduction by asexual means is strobulation. So, very good example for strobulation is jellyfish. We could see jellyfish in the oceans, right? Very beautiful organisms. So, one species of jellyfish is Aurelia. Let us see the life cycle of this jellyfish. See, this is an adult jellyfish. It lays eggs. The eggs gets matured at young stage arise, and after that adult stage. In this adult strobila, look at these structures. These are called transverse fissures. That means in the body of that organism, in the adult strobila, layer by layer, the cell division occurs. They are called transverse fission. Okay. So after a certain time, what happens? Every layer gets detached like this. This is called ephera. Every individual layer from here, it gets detached and it develops a nucleus and it develops into a adult jellyfish. So from one young adult strobila, many jellyfishes, they will evolve. Okay. This method of reproduction is called strobulation. Now, we have seen many different types of fission. Binary fission, multiple fission, sporulation, strobulation, like this. So, what is the advantage of this method? Why organisms choose this method of asexual reproduction mostly? Because this method is very, very rapid. So, for example, you take Escherichia coli, E. coli. It's a bacteria. Its generation time is just 8 hours. In 8 hours, one cell becomes 2 cells. Okay. So, similarly, if some 100 cells are present, in one day, millions of bacteria will be produced in a particular environment. Okay. So, this allows the organism to crowd out other organisms that reproduce more slowly. For example, in order for this unicellular organisms to survive, to exist, to dominate, compared to the other higher cellular organisms like plants or animals or human beings, they have to multiply, reproduce fast, right? Even in our body, we have millions of bacteria in our body. They are called normal flora. Okay. the number of normal flora are very high compared to our own body cells. Okay, how this is possible? Because these bacteria, they reproduce very fast. Okay, so similarly for any organism, for example, we are getting many different types of diseases by bacteria or parasites. So that very minute microscopic organism enters our body and it's reproducing very much faster. That's why we are getting disease. That's why it's able to kill us also, right? So in order to compete with the higher organisms, these organisms should reproduce very fastly. So that is the major advantage of this fission mode of reproduction. Okay guys, so let us have a quick recap of what we have learned in this lecture. Reproduction, we can classify into two types, asexual and sexual method of reproduction. Asexual reproduction has fission, budding, fragmentation, sporulation, vegetative reproduction, gemule formation, regeneration, and agametogenesis. And this fission, again, we can divide into four types: binary fission, multiple fission, sporulation, strobulation. Again, based on at which angle the binary fission occurs, we classify them into four types: simple irregular binary fission, transverse binary fission, longitudinal binary fission, and oblique binary fission. Okay, hope you understand this lecture. Our coming up next lecture is about the other types of asexual methods of reproduction. If you do like this lecture, please give a like and share it with your friends, and don't forget to subscribe our channel NEET Biology. Meet you in another lecture. Until then, take care. Thank you.